is Dr. Philip Kim, so from University of Toronto. Philip is a professor at University of Toronto, co-founder and CTO of Fable therapeutics, and a specialist in machine learning and computational biology. His group combines modern AI methods with high-throughput experimental data to enhance drug development and therapeutics, and then he's going to talk about ML methods for protein and peptide design. There you are.
Okay, thank you. Yeah, thanks for the invitation. It's great to be here, and it's great to be at a meeting with so many different talks. So I'll talk about something completely different. I'll talk about protein design and peptide design, and it's great to be exposed to some more biology because I tend to, just look at proteins these days. Okay, as mentioned, so I will mainly talk about my academic work. As the introduction kindly mentioned, I wear many hats, and among others, I run a biotech company. Right. Okay, so so the machine learning revolution has really made my life very exciting, and so the last 5, 6, 7 years in protein science have been extremely exciting. And what is the progress has really been tremendous. Has really been astounding, right? So I've been doing this for a fairly long time, right? And after several decades of pretty steady but very incremental progress, we have seen like 5 years of several revolutions happening, just one after the other, and things that were absolutely science fiction just a couple of years ago, we can do that now, right? And pretty much everybody can because most of the software is available, right? So we now live in this time where this dream, and it really was a dream, of making biologics or protein-based therapeutics on a computer is basically reality, right? We're not totally, it's not everything, and it's not perfect yet, right? And we're still working towards making it more perfect, right? But it is definitely reality, right? And so, I want to give a bit of a shout out to what made that possible, right? What made it possible is a fairly large body of data that has accumulated over the years, and most notably for my purposes in the PDB, right? So there's hundreds of the protein structures in the PDB that form a fairly high quality set of training data, right, that we can use. I want to give a brief shout out to Moore's law, right? So I guess we're going to talk about this now, but I just want to remind everybody that every one of us walks around with a supercomputer in our pocket, right? This used to be a supercomputer, right? Like just when I did my PhD, this was a supercomputer, right? And then of course we'll be at the advent of machine learning methods, and I will do a bit of a walk through what happened in the last 5 years, 6, 7 years, right, in the protein world, right, and I will claim a bit of personal credit, whether we deserve it or not, for some of the advances, and right. So what happened? What had to happen for all these great advances of machine learning to make an impact in protein design was a way for us to encode the data in a way applicable to machine learning, right? And so very early approaches, and I still remember talking about it with colleagues and students about how one would use, and back in the 2015, 2016, right? AlexNet was already a thing. CNNs were all the rage, right? And how do we do CNNs on proteins, right? Protein structure, right? And so the early approaches, and there's papers out there that do that, right? The main problem is you have a protein structure, which is a 3D object, right? A protein is fundamentally a 3D object, right? And then CNNs operate in 2D, or like language models like RNNs back then would operate in 1D. And then how do you do with 3D? So the simplistic approach was to use volumetrics, right? So you, instead of pixels, you do voxels, right? And then you make 3D space into little boxes, right? And then you do your CNNs into little boxes. And that never really worked, right? So that's why you never heard about it, right? That never really worked, and we played around with it, and never really worked very well. And so one breakthrough came really when people started using graphs, and my group did some of the early work in this graph-based work. So essentially, you can break down your protein into amino acids as nodes, right? And then you encode some information in the nodes. You can encode some information in the edges, and that has certain advantages. Like you don't have to discretize, and you get certain mathematical properties, like you get invariance, right? So so it's just nicer for the computer to deal with this in a graph setting. This has evolved now several times, and one evolution was that all the rage used to be equivariance, and then earlier this year AlphaFold3 showed up, and they proved that equivariance, you don't really need equivariance. And if you're interested, then I'm happy to have an in-depth discussion with you on equivariance and why I think it's still a good idea, but anyway, that's a separate topic. So these graph methods really paved the way for a lot of what came after. And we, in our lab, we applied the graph methods. We used the graph methods to develop the first inverse folding, so the first protein sequence design method. So you have a protein structure and you encode it as a graph. You train a model to fill it with amino acids, and then these amino acids will then fold into the protein that you want to. And we make use of the PDB, and we make use of graph convolution, and we could show that this really works at regenerating real proteins. So this was effectively the first method to do fixed backbone design with machine learning. We could show back then, that was the pre-AlphaFold world, but we could still show that all the proteins we design in all alpha, all beta, alpha/beta, they make real proteins. We could make them proteins in the lab, and we could show that they are really folded proteins, right? And back then in the dark ages, in 2019, 2018, when we did this was amazing, right? The computer comes up with a protein sequence that actually folds, right? This was like really, right now it's really trivial, but that was kind of an eye-opening moment for me to see that the machine learning in protein design and protein structure really has a role to play, right? And I will note that inverse folding is now considered to be a mature. These approaches now have now matured, right? And the newer approaches are protein MPNN is probably the one that most people use. Is conceptually it's very similar. It's basically the same protein solver, but it is much better engineered, and it is probably the method to use, right? And there's more innovation. There's a bit more innovation happening, but it's largely considered to be a mature problem. Okay, so that's for protein sequences on given a structure. Now we want to design the structure, and so we want to do de novo design, and for that we turn to diffusion. And I'm a little surprised to have had a full day of AI talks and nobody has mentioned diffusion yet. So I'm going to mention diffusion now. And so people, if you haven't heard about it, I'll give you the 2-second explanation. The 2-second explanation is that you can train an AI and a neural network on learning data from the noise in a stepwise manner. That's not really true, but anyways, that suffices as a quick explanation. And diffusion is an interesting thing that it came out. Diffusion came out in 2015, so like a really long time ago. Nobody really took notice until the first diffusion method started to beat GANs at image generation. That was in 2020. And ever since then, diffusion has been all the rage, and everybody has probably seen DALL-E or Midjourney or Stable Diffusion, or all these diffusion-based image generators, but they work really well. So then a new student came to my lab, and he said he's really excited about this diffusion thing, and he wants to build a protein design method using diffusion. And I was like, oh, that sounds like a great idea, but I mean, I told him basically we have to do it tomorrow. And we have to publish it the day after tomorrow because otherwise we're going to get scooped. And so he then went on his way, and we did actually very well. So we were the first validated diffusion model out in the summer of 2022. Before other people like a biotech company Chroma or RF Diffusion or the Baker Lab came out of Fable after us. So what we did essentially, we took again this graph-based method. And the good thing about a graph-based encoding of the structure is you can actually design it as a matrix, right? And then what we can do then is we use these 60 matrices, right, to get a fairly detailed encoding of the protein backbone structure, right? So you can do distances and angles, right? And we can then train a machine learning method to essentially generate these sort of matrices, right, in a diffusion model, and then you get out the protein backbone, and then you can use a sequence design method like protein solver or MPNN or Zeta to then fill it with sequences to get a real protein, right? And amazingly enough, back then it was amazing. Back then that was really like astounding. Amazingly enough, that actually works, right? So you're getting out these sort of proteins, right? And they look like real proteins in sort of any way we can tell computationally, and some of these proteins are really new in the sense that they're very different from anything you would have in the PDB. So they're proteins that nobody's ever seen before, and they really exist. And you can actually make them in the lab, and you make some of them in the lab, and you can show that they're nicely alpha helical, alpha fold sinks, they fold this way, and you can melt them, and they have a native melting curve, like a nice two-state transition, so they're really well-behaved proteins, right? And yeah, so there you have it. So this really works, right? And we can now, and everybody can now, with protein SGM here, or with our diffusion, or with Chroma, or with Genie, or with there's now multiple protein diffusion models out there that many of them work very well, right? You can now make real proteins, real new proteins just on the computer, right? And that really works, right? And that's just in 2022, this was absolutely mind-blowing, I think. And now it's 2 years later, and it's like completely trivial, right? And I think that's a good gauge of how fast the field has moved, right? That this is true, right? Okay. So now we want to do something useful, right? And as mentioned, I did co-found a company, and I run the machine learning part of the company now, and we make antibodies, right? And we make antibody therapeutics specifically. And so and at Fable, we build I would say a state-of-the-art diffusion model to do structure and sequence design for antibodies, and we can show in these benchmarks here of sequence design and structure design. We outperform the public state-of-the-art by a fairly healthy margin, right? And we can do now antibody candidates that we're now advancing in the therapeutic pipeline. And depending on the problem setting, we can be very accurate, and we can get when we design antibodies and we test them in the lab. Depending on the setting, we get hit rates up to 60% or something, right? So as I mentioned, the dream is very much reality. It's not perfect yet, but it definitely works, right? And there's not only Fable, but there's multiple companies who are doing this. Of course, Fable is the best company, obviously, right? But this is now really happening, right? So the future of biologics discovery, of antibody discovery, is definitely computational and AI driven. Okay, I want to switch gears a slight bit. I'm gonna cover the next two topics very briefly. So I wanna talk a bit about dynamics, right? So that's what I'm academically excited about, right? So we wanna, and that's especially true for peptides or peptide microcycles. So structure design and structure prediction, that's old hat now, right? So that's kind of solved. And so now we want to design, or we want to predict the dynamics, so the conformational energy landscape of a peptide of a molecule, right? And so those of you who are physics or physical chemistry inclined, and there are always some people like this in the audience, you may know that there's some conformational energy landscapes that we can show like in this like 2D cartoony fashion, right? And of course, there are methods to sample the conformational landscape using a trajectory-based method like MCMC or molecular dynamics, right? But the main problem is that these often, to go from one state to the other, often take a long time, right? So that's the problem. And so what we want to do, what we want to do really is we want to get the full energy landscape out in one go, right? So we want to essentially predict that whole thing. So structural prediction is predicting this one thing here, this one state, but we want to predict the whole landscape, right? And so to do that, we can turn to machine learning, And what we want to do effectively is this is the conformational energy landscape, right, in a 1D setting. And we want get a neural network that'll start with a Gaussian and then convert this Gaussian into an energy landscape, right? And that's a neural network. And by the way, that's kind of exactly what diffusion is doing, right? Diffusion converts a Gaussian prior into your target distribution, right? And so we did that essentially. And since we're talking about explainable AI. You know, the part where we're sort of closest to explainable AI is the PepFlow. This method here is it consists of a hypernetwork and a real network. So this network here actually does the prediction of the conformations, right? And it's a relatively small network of only 100,000 or so parameters, right? But sort of the heavy lifting is done by the hypernetwork, and this hypernetwork predicts the parameters of the smaller network. And then this put together then, and it's altogether a pretty complicated approach, will then generate conformations of all atom conformations of peptides. And so it works really well. So we're getting I would say state-of-the-art results for single structure prediction. But we also get different conformations out, and we're doing quite well at both precision and recall of NMR conformations, right? So of real conformational energy sampling from experiment, right? And as you can see here, right, if you compare to MD, we are of course much faster to sample more conformations. You can see in MD, you will always have this trajectory-like approach, like all the samples you're getting are similar to each other. With the PepFlow samples, you're sampling directly from the energy distribution, and you're getting a different minima out right away, right? And so this is of course an advantage. And so now we're using PepFlow in a binary design manner, right? And the big prizes, and I'll skip that. The big prizes to do docking or to do binary design directly. So what we're doing now is Pepflow++ is using this method to generate bound peptides as binders or then in the future as therapeutics, right? And in the last couple of minutes. I want to do because we had a couple of great talks about gene regulation, right? So I want to talk a tiny bit about gene regulation, and what we're focusing on is C2H zinc fingers, and C2H zinc fingers are of course are the most common transcription factors in the human proteome, right? So these are C2H zinc fingers. They used to be the main technology people use for gene editing or for all sorts of things, and that because they are highly engineerable, right? That was before CRISPR came around, right? And CRISPR, of course, sort of kind of totally blew them away because everybody can use CRISPR and you can make a guide RNA and you can edit your gene you want, but zinc fingers still have certain advantages, and they're human proteins. So there's many zinc fingers. There's 7,000 zinc fingers in a human proteome. So they're less immunogenic than CRISPR, which is a bacterial protein. They are pretty small, and they can be used to bind any DNA sequence, right? So for instance, especially for in vivo therapeutics, right? So if you want to use it on yourself, if you want to inject yourself with a transcription factor, you probably want a human protein that's non-immunogenic, because your body is not going to like it if you inject yourself with CRISPR, right? And so that's why zinc fingers are still that's why zinc fingers have advantages over CRISPR in this in vivo setting. The main problem is that predicting zinc finger interactions was really difficult. And you couldn't. It was previously impossible, essentially, to generate a code that tells you, given a certain amino acid sequence, what are the given a certain DNA sequence, what are the zinc finger amino acids that will bind it tightly, right? So that DNA to amino acid code was impossible to solve, and that has to do with the complexity of zinc finger DNA interactions, and I won't get into that, but so to make a long story short, my collaborator Marcus Noyes at MOU generated a very large set of data so billions of data points on zinc via DNA interactions, and then we use that with a I would say reasonably smart setup of using a transformer, but in a hierarchical transformer, to essentially convert DNA targets into zinc finger amino acid sequences that will bind this target. And so this is a classic transformer problem. A transformer goes from space A to point B, right? So the transformer fits very well. And then lo and behold, this works really well, and it blows everything else out of the water. What was there before? That's not that difficult because everything out there was really bad, and it works very well at getting you the right sequences. And we tested it in a way to reprogram transcription factors, right? To make transcription factors that can either repress or activate any gene you want to. And here we did it against DPH1, and you can see here. So this is the control, and these are the ones. This is the control, and this is all the things we tested, right? And you can see that every single one of them works at repressing the control DNA, and then 3 of them work really strongly. And we can do it for activation too against a different gene. And you can see here this is the control. We get activation of a background every single time, and some of them we get very strong activation, right? So it really works. And I'll skip the rest. I just want to leave you with this really works, right? Is we can get single single target specificity, and with a bit of engineering we get very strong and very specific zinc fingers. And I want to close here, almost on time. This is my lab. This is my funders, and as I mentioned, I wear many hats, but some of these are some of that way. Okay, thanks, and thank you, and I'll take questions if there are any.
Questions in the room?
Thank you for the talk. That was great. I was wondering if you had any thoughts on how protein language models compare to the graph-based methods, and if you thought that the graphs add an element of interpretability that the language models lack, perhaps.
Yeah. So there is now that sort of philosophical debate around whether we can use tokenization in a language model setting versus continuous representations of the protein structure, right? And so most of the design methods right now work in a continuous setting with diffusion, right? And that is sort of, and that may well just be sort of a inherent bias of the people doing it, because I think that makes more sense, right? And I think many people like me will probably think that makes more sense. But tokenized versions like ESM3 is a tokenization, right? And I mean, you can say that it's total overkill because they put in 4,000 structure tokens right in, and then it works really quite well, right? And they get a good accuracy. They come very close to AlphaFold3, I think, and some of the accuracies, right? So there are some folks in my lab now who are playing around a bit with this tokenization, and we do find, I mean, there's been other tokenizations, right? And we do find that ESMC seems to work the best. I think it's still the jury is still out whether which will win in the end, right? I mean, you can argue, you can make arguments, right, that tokenization at some point, with scaling, you might win, right? But right now, I think that the continuous models are still better. And to interpretability question, right? I mean, in the protein structure world, I'm a bit ashamed. I'm a bit ashamed to say this in this explainable AI conference, but we haven't been overly concerned with interpretability so far, right? We've just been very engineering, like just get it to work, right? I will say though that there is a bit of a debate in the field on on that, but I strongly believe that, for instance, that AlphaFold learns some part of physics, of protein biophysics, right? And sort of part of the metrics you can get on AlphaFold correspond quite nicely, I think, to an energy function, right? So I think that's a debate. Some people would strongly disagree with me, but I'm speaking here, so I can say that. So in that sense, you have interpretability in that sense, right? You don't have interpretability of the actual weighted parameters because there's obviously yeah.